Hello and welcome to Psyched, the show where we explore psychedelics through social, economic, and political perspectives.
Our next speaker is Nick Milne. He'll be talking on accessing psychedelics without exploiting natural or cultural resources, yeast fermentation as a scalable and cost-effective production method for psychedelic molecules. Nick Milne is a fungal biological engineer and co-founder and CSO of Octarine, a synthetic biology company developing yeast-based production platforms for cannabinoids, psychedelics, and their novel improved derivatives. Nick's research focuses on understanding how and why organisms produce specialized molecules, and through genetic engineering, how this can be exploited to improve human health. Driven by the growing mental health crisis, Nick began investigating the metabolic pathways various organisms use to produce psychedelics, and by transferring the genetic information into yeast, demonstrated a viable production method for psilocybin and a range of psychedelic tryptamine derivatives. Nick, thank you so much for joining us and welcome to Psyched.
Thanks for having me. Let me just share my screen. Okay. Okay, thanks for having me. Hi everyone, I'm Nick Milne. I'm the co-founder and CSO of Octarine. Really excited to be here today to tell you all about our company and what we're doing to access psychedelic molecules without exploiting natural or cultural resources, all using my favorite microorganism, Saccharomyces cerevisiae, which is perhaps better known as baker's yeast or brewer's yeast. To quickly introduce you to the company, we're founded in October 2018. We're based in beautiful Copenhagen, Denmark. And what we do is we use yeast fermentation to produce psychedelics and cannabinoids. We also develop enzyme modification platforms to modify these molecules to improve structures with improved properties. To quickly introduce you to the team, there are two co-founders, both myself and Nataji Galaj. Nataji is a plant biochemist and an expert in cannabinoid biosynthesis. I'm a fungal engineer and fermentation scientist. And collectively, along with the rest of our research team, we have a lot of industrial and academic experience engineering yeast to produce a range of high-value molecules and scaling up their production in fermentation. Our management team and our scientific advisory board is a nice, healthy mix of experts from the industrial biotech industry, as well as the drug discovery industry. And our team has backgrounds from companies such as Novo Nordisk, Novozymes, Evolva, Lundbeck, Pfizer, and even a university professor. So we're all here today, I think, because we understand the slide, and we're all really excited about the potential for psychedelics, particularly to treat a range of mental illnesses. As somebody who's spent a lot of time on a range of antidepressants and various other drugs, it's clear to me that the current model for treating mental illness doesn't work, and we drastically need a radically new treatment paradigm. And it's my hope, and I think everyone here would agree with me, that psychedelics could be this new treatment paradigm. And when we talk about psychedelics, we're actually referring to a range of different molecules produced by all sorts of different species. And as you can see, there's a really healthy mix of fungi, there's cacti, there's plants, there's sea sponges, there's toads. I've included cannabis in this picture, while not technically a psychedelic, because we work with it at Octarine, it goes on my list. And if we accept that sometime in the future, psychedelics will be approved as therapeutic treatments, and one day you'll be able to go to the doctor and get a prescription for a psychedelic therapy, we need to think how we're going to meet this expected global demand. And one thing we should consider is, where are we going to get these molecules from? So to facilitate pharmaceutical application of these molecules, there's a few things we need to consider. Firstly, the molecule needs to be produced relatively cheaply. If it costs too much to make, then this cost can be passed on to the patient. And if we want to ensure equitable access to these molecules, we really need to make sure that it can be produced for a feasible cost. We also need to produce a lot of these molecules. One example from the cannabis industry, it's expected that just the expected pharmaceutical demand for CBD in the next 5 years is going to exceed 350 tons annually. We're going to need a huge amount of these molecules. And we also need to produce them in a stable supply. This means that the supply chain needs to be intact and stable. And it also means that the production process needs to be stable, and there needs to be very minimal variation between each batch of production. It needs to be extremely high purity. So we're talking 99.99999% pure. And while not strictly required for getting these products to market, we really should also consider the sustainability of producing these molecules. So where will we get them from? The most obvious place to start is to just extract it from the native host. So extract psilocybin from Psilocybe cubensis mushrooms. Unfortunately, in most cases, this is not a very good method. It's usually pretty expensive, primarily because these molecules tend to accumulate in very small quantities in the native host, meaning you need to cultivate quite a lot of the organism to get enough. This also makes scalability an issue. Going back to the CBD example, to meet the 350 tons of CBD that will be required, there's actually not enough land on Earth to meet this demand via extraction from the cannabis plant. So this is not going to be an effective method. Stable supply is often difficult. These the species that produce these molecules can be difficult to cultivate. They also grow in specific conditions. They also grow in specific seasons, leading to a lot of variability. And extracting these molecules from native hosts in general, it's often difficult to achieve a stable supply. Very small differences in the way these species are cultivated can lead to big differences in the final product. It's difficult to purify the molecules. These species that produce psychedelics, they produce all sorts of other things, and it can be very difficult from a purification point of view to separate your psychedelic molecule of interest from all the other things that the organism produces. And in terms of sustainability, while there are plenty of examples of sustainable production of natural molecules from native hosts, unfortunately, this is not always the case. And we're unfortunately starting to see this already for psychedelics, not only from from an environmental point of view, but also from a cultural and ecological point of view. We're already starting to see declining populations of some of the species that produce these molecules. And of course, the other thing to consider is that many of these molecules and the species that produce them are sacred to many of the indigenous communities around the world. And if we want to bring this to the world, it can't come at the expense of the indigenous communities who often were the ones to teach us about these molecules. So the next place to look is chemical synthesis. This is a very common method. It's been used for hundreds of years. And in fact, of all the pharmaceutical drugs derived from natural products, almost all of them today are chemically synthesized. And it's a really good method, I have to say, but it's not perfect. It can still be very, very expensive. As an example, psilocybin produced by chemical synthesis today costs about $7,000 per gram. I'm sure this will come down in the future, but it's still quite expensive. And it can be very difficult to scale the production of these molecules. Chemical synthesis is really good for simple molecules, but as the molecules start to get more and more complex, it gets more and more difficult to scale. Cannabinoids, for example, are pretty complex molecules, and as such, it's not really feasible to produce them by chemical synthesis. An example from psychedelics is ibogaine. The class of molecule that this molecule comes from is actually described as being the most complex natural product ever discovered. And as such, chemical synthesis will not be a scalable method. While you can achieve a stable supply and very high purity using this method, it really falls apart when we think about the sustainability. And the big ugly secret of the chemicals industry is that approximately 90% of all the commercial chemicals available today are derived from crude oil or other non-renewable resources. So this includes all of the pharmaceutical drugs we use, but all of this, a lot of the ingredients that go into producing our food, and all of the bulk chemicals that go to making all the products we enjoy in our daily lives. Most of it isn't sustainably produced. So what Octarine proposes is to use yeast fermentation to produce these molecules. Just to be clear, yeast is this microorganism that produces beer and wine and bread and all those other things we love. And it's really kind of a middle ground between native extraction and chemical synthesis. You can get all the benefits of a natural production host with the benefits of an industrial scale production process. And to explain why we should produce psychedelics by yeast fermentation, I have to first tell you the story of how yeast turned from this wild organism that grows primarily on the bark of trees into this organism which really thrives in an industrial environment. I first have to tell you, the story is actually how we got to this point is actually the story of how humans learn how to get drunk. Humans have been producing alcohol for about 4,000 years. And back 4,000 years ago, what they would do is produce some sweet liquid. They would leave it exposed to the air, and they would hope that some yeast species would fall in and turn it to alcohol. Sometimes this worked. Sometimes a bacteria might fall in and turn it into something awful. Sometimes a bacteria might fall in and actually turn it into something that's going to make you sick. But this trial and error approach kind of persisted into the 16th century. By this stage, brewing beer was a legitimate profession. And what the brewers started to realize was that if they managed to produce a really good batch of beer, what they could do was leave a little bit of the beer behind and use it to start the next round of fermentation. And when they did that, they found that they consistently got a good tasting product. Now, of course, what they were doing is leaving a little bit of that yeast behind and using it to start the next round of fermentation. But unintentionally or unknowingly, what they were doing is selective breeding. And one of the first examples of that, they were selecting for yeast that could produce better tasting beer. And as fermentation technology continued to evolve, they started to select for yeast that could do other things. They started to select for yeast that could produce alcohol more efficiently or more quickly. They started selecting for yeast that could tolerate the harsh conditions typically found in a fermentation environment. And they started selecting for yeast that could tolerate, that they were resistant to infection and other things like that. Fast forward today, and today's modern yeast is this very tolerant species that just thrives in a large scale environment. And now it's not only used to produce beer, but it's used to produce a range of other products. It's used to produce the biofuel that might go into your car. It's used to produce the enzymes that you might find in your laundry detergent. It's used to produce a range of pharmaceuticals, such as human insulin. It's even thought that the next coronavirus vaccine will be produced in yeast. So there's a long and very progressed history of using this organism to produce useful products. And so why produce psychedelics by yeast fermentation? Going back to my initial requirements, it's very cost effective. The bulk input is sugar. It costs about 20 to 50 cents a kilo, meaning we can get very low cost of production. It's massively scalable. There are bioethanol plants as well as beer brewing fermentation plants all around the world. This method can be scaled anywhere around the world to any scale. With yeast fermentation, you achieve a very stable supply. A good way of thinking about this is wherever in the world you are, a Heineken tastes pretty much exactly the same, even though it's brewed in different fermentations. With yeast, you can get a very consistent product. The great thing about yeast is that it will produce your molecule and then it will secrete it into the surrounding media, which makes purification really easy. You can just separate the yeast from your product and half the job is done already. And one of the big reasons why there's a lot of interest in using yeast fermentation these days is because of the big advantages in sustainability you can get. Now, because psychedelics aren't produced on a large scale at the moment, I can't really give this comparison, but cannabinoids are produced by large scale today via extraction from the cannabis plant, so I can make this comparison. And as you can see, producing a kilogram of CBD from cannabis versus a kilogram of CBD from yeast fermentation has huge increases in sustainability. And the thing to say about cannabis cultivation is it's actually surprisingly unsustainable. It requires a huge amount of land. It's typically cultivated indoors, meaning you need a lot of energy. It uses a surprising amount of water and a surprising amount of CO2. CO2 goes into the extraction process. It's also often pumped into the greenhouses to make the plants grow quicker. So with yeast fermentation, we can have huge boosts in sustainability as well as ecological sustainability. The other big advantage, of course, is that we can drastically reduce the production cycle. For cannabinoids, this goes from 6 months to only 2 weeks. So what Octarine does is it engineers yeast to produce cannabinoid and psychedelic molecules. To do this, we identify how native species produce these molecules. We identify the genes and enzymes involved. We transfer all of this genetic material into yeast, enabling the yeast to produce the molecule of interest. Then we can scale up the production and fermentation. So the way to think about this is yeast is kind of like a miniature cell factory. Sugar goes in and out comes your product of interest. So some examples. We recently published our work on engineering yeast to produce psilocybin by fermentation. So we identified how the Psilocybe cubensis produces these molecules. We transferred this into yeast. We played around with the native yeast genome a little bit. And in 1 liter fermentation, we could demonstrate quite good production. This was featured in a few news articles, and I have to say, so far, the highlight of my scientific career is being featured in High Times magazine. But the numbers you should be interested in are the ones on the bottom right. This proof of concept study took about 6 months to achieve, and already we can have a production cost of around $20 per gram. The big thing, though, is that in 2 years, we'll have this well below $1 per gram. As you can imagine, this is a huge cost saving. The other thing we did, which I think is really cool, is we could show that by making small tweaks in the yeast genome, we could tailor the production to a range of psilocybin derivatives and intermediates. So psilocybe mushrooms, they produce a range of intermediates and derivatives that we really know nothing about, primarily because they accumulate in such small amounts that we've never really been able to extract enough to test it in any way. In the study, we could show that we could produce all of the known psilocybin derivatives, such as baeocystin and aeruginosin. The other cool thing we did is we took a gene from the melatonin biosynthesis pathway of cattle, introduced it into our yeast strain, and we could demonstrate the production of a completely novel molecule that doesn't exist. This molecule half resembles melatonin and half resembles psilocin. And what's cool here is that we can take genes from different organisms, put them into one species, and have the production of a completely new molecule. Since this paper came out, we've also demonstrated the production of a range of other psychedelics from other species. I wish I could tell you more about this, but at the moment I have to keep my mouth shut. Hopefully we can say more about it soon. But this kind of brings me to the next part of our talk. I've talked about how we're producing natural molecules more efficiently. But the question we asked is, why should we stop at only producing natural molecules? Why not go a step further and make new molecules with improved properties? This kind of approach is not new. Actually, it's very common in the drug discovery space. And medicinal chemists, they quite often take a natural product, a natural molecule, and they use chemical synthesis to derivatize it in a bunch of different ways, to create a library of new molecules and to hopefully find one that is better. That method has been used for around 100 years and it works pretty well. But what Octarine proposes is to use enzymes to do this instead. And by using enzymes, we can offer some pretty unique advantages. Firstly, we can have what's called a one-pot production. which means that we can go from sugar as our bulk substrate and go all the way to our improved derivative in one step. This helps with the production and the purification. Enzymes have evolved to work under pretty ambient conditions. So we don't need things like high temperatures or pressure or expensive catalysts. These just work under normal conditions. And perhaps the biggest advantage is the incredibly high specificity you get with enzymes. We've developed enzymes, for example, that can take this indole skeleton and they can very specifically add a hydroxyl group at the 4' position or the 5' position, but not either. This means that we can produce these molecules with 100% stereospecificity, near 100% yield, and all under ambient conditions. And this is something particularly interesting because this is something very hard to do by chemical synthesis and really shows the potential of our yeast production platforms. So we've been using these enzyme platforms that we've developed to improve the properties of cannabinoids and psychedelics. At the moment, I can't talk too much about our psychedelic derivatives, but I can say a bit about our cannabinoid derivatives. If you know anything about cannabinoids, you know that they're completely insoluble in water. They degrade under pretty ambient conditions. If you expose CBD to oxygen, it turns into this purple quinone product. If you expose CBD to acidic conditions, it degrades into THC, which is obviously a big problem. And these molecules have a very low oral bioavailability. We have developed enzymes which can convert natural cannabinoids into improved derivatives. We've created derivatives that are completely soluble in water, that are completely stable, even under extreme conditions, and derivatives that have a big increase in oral bioavailability. And the important thing here is that the derivatives that we're making, it's infeasible to make them by chemical synthesis. So what we're doing is we're expanding the chemical diversity beyond these traditional methods, enabling access to new kinds of molecules. So to wrap up and to talk a bit about the business model, we have a lot of clever scientists doing a lot of clever things with psychedelics, but there is a business model behind this. At Octarine, we develop cell factories for high-value molecules, mainly cannabinoids and psychedelics. We develop enzymes which can convert these products into new molecules with improved properties. And we also do the preclinical studies or the product development, primarily to demonstrate the potential of these molecules. We work with partners to put these molecules into products and bring them to market. We're primarily interested in therapeutic applications, but we have, for example, been approached by a few cannabis consumer companies, for example, that are interested in extending the shelf life or water solubility of cannabinoids. So we're also looking at consumer applications. So we are very interested in talking with biotech companies who are looking for a stable supply of some of the psychedelics in their pipeline. We're interested in talking to biotechs who perhaps want to investigate new derivatives of some of their molecules. We're also interested in talking to anyone who has a good consumer application for some of these molecules. So I'll stop here. I'll thank you very much for your time, and I'm really happy to take questions.
Hi, Nick. Thank you so much for the chat. Really appreciate it. Let's see, we got a few from the audience. One of us asking about if you could talk a little bit about entourage effects of molecules that occur in small quantities in natural sources. They give a little bit of context talking about, we know how terpenes modulate cannabinoid effects in the body, but it's looking like that this happens with serotonin agonists too, biocysteine and psilocybin species, for example. How will you address this?
Yeah, so, I mean, the entourage effect is quite well documented in cannabis. It's thought that maybe this is occurring with psilocybin mushrooms, but it's really too early to know. I think one of the main reasons we don't know is because we just can't study these molecules. We don't really know. The thing about an entourage effect, at least in my opinion, is that it's the action of a few molecules acting together. So it could be used as an argument to maybe say we need to look at whole plant or whole mushroom extracts. But I think if you're thinking of pharmaceutical applications, this kind of approach is really difficult to get through FDA approval, for example. So I think it's going to be much easier to get a single molecule through approval. This is certainly the case with cannabinoids The molecules, the psychedelics currently in clinical trials are all with a single molecule. I think this will be the way that pharmaceuticals will develop.
Mhm. Could you talk a little bit about the N-acetyl-4-hydroxytryptamine? Is it psychoactive? Isn't it easy to make by chemistry?
So to be honest, we know nothing about this molecule. What we wanted to do was just demonstrate that you could take a natural organism, you could take natural enzymes, combine them together and make a completely new molecule. This is kind of the direction that Octarine's going, particularly with cannabinoids and also with psychedelics. We're not saying that this particular molecule does anything, but it does demonstrate that we can use this cost-effective, sustainable production process, not only to make natural molecules, but also to make new ones as well. And I think there's a lot of potential there.
Are you all just focused mostly on the generation of IP and kind of these new, research approaches to development, or are you planning to get into products and kind of consumer-facing goods at some point?
So we're pretty much strictly a B2B company. So we are mainly looking at other companies to help put these into products and bring them to market. I think very early on, we realized that we can do a lot of things with this technology and we can't do everything. So we'd prefer to work with a few different companies in different ways. And, we have a few partnerships ongoing at the moment in quite a few different areas, which I think really demonstrates the potential of the technology.
Mm-hmm. Could you tell us a little bit about the intellectual property landscape in the biosynthesis space? Is it crowded? Is the room? Yeah, just some of your thoughts on it.
So with psychedelics, it's a very new field. So there's certainly a lot of room in the IP landscape at the moment. For cannabinoids, the biosynthetic production it's been around for a few years already. As such, it's really crowded. Also with biosynthetic production, the IP protection you can get, for example, producing psilocybin in yeast, it's kind of hard to get a strong IP position in there. So Octarine's focus is more on the novel molecules or the enzymes that can catalyze these novel molecules. That's kind of where I think a lot of the value in these business models comes.
Mm-hmm. And could you talk a little bit about Octarine, and there's other companies in the space that approach kind of the work that you're doing with yeast-based biosynthesis of either cannabinoids or psychedelics. Could you talk a little bit about how Octarine is different from those other companies?
Well, I mean, to be honest, there's a range of companies making a range of molecules by yeast fermentation. I'd be lying if I said we invented the technique. It has been around for a long time. It's been used to make nutraceuticals and biofuels and all of that. I think Octarine's been around for a little while, and so we're a little bit ahead, I think, on the psychedelics production. And this work that we described is about a year old now, and we've progressed quite fast since then. I think the big differentiating factor is the enzymatic derivatization, and I think it's kind of the thing we're really excited about. This technique that, the molecules that we can make, I think, are very hard to do chemically, and it's particularly very hard to scale up chemically. So I think that's kind of our differentiating factor, and I think it's what we focus most on, particularly in the future.
Mm-hmm. How do you make a yeast strain secure? What if it gets out of the lab?
I get asked that all the time, actually. When the psilocybin paper came out, I think I got maybe 50 requests if they could send me the strain. There's a pretty strict biosecurity protocol, particularly here in Denmark, and actually all around the world. There's a range of security that you need just to have a genetically modified organism. The risk is definitely there. If somebody steals our yeast strain, I don't think we would ever get it back. I think the answer is that governments around the world have pretty strict protocols in place to work with these molecules in our lab, and even just to generate genetically modified organisms. We need to jump through quite a few regulatory hoops. We need to demonstrate that for example, that we have proper security. I don't think there's ever been an example of a yeast strain being stolen. As far as I know, there's no example of a yeast strain being let loose in the wild. So I don't think it's a big issue.
Yeah, and one last question. Does the yeast turn blue like the mushroom?
I probably shouldn't say. I think maybe we're going too a bit far, but it's something we've looked into because the mushroom does turn blue. It turns blue on exposure to oxygen. It's basically a oxidative reaction. We have seen it turn blue, but under specific conditions. That's all I can say.
Okay, awesome. Nick, thank you so much for your time. We really appreciate it, and looking forward to talking offline.
Great. Thanks for having me.
Be well. Bye-bye.